Bueno, vamos a dar comienzo entonces a este evento. Buenos días a toda la audiencia. Muy bienvenidos a la inauguración de este segundo ciclo virtual de conferencia o Virtual Open Meeting, como comúnmente lo llamamos VOM. Como ya hemos mencionado en otras oportunidades, la realización de la edición 23ª de SINAQO en forma virtual nos ha motivado para emprender nuevos desafíos. Por esta razón, hemos implementado otras modalidades de comunicación y discusión que espero que sean del interés de todos. Ellas son la realización de 3 ciclos de conferencias virtuales, de los cuales ya ha transcurrido el primero de ellos, el 24 de junio. Hoy estamos inaugurando el segundo y el tercero estará previsto para el 22 de septiembre. Otra de las innovaciones que hemos implementado para esta edición ha sido la creación de un consejo asesor tecnológico integrado por investigadores y empresarios de la región. Tanto en la formación del Consejo Asesor Tecnológico como en la ejecución de este ciclo, lo hemos organizado conjuntamente con el Ministerio de Ciencia y Tecnología de la provincia de Córdoba. Por eso hoy en la inauguración me acompaña el doctor Gabriel Raya Tonetti, secretario de Articulación Científica y Tecnológica del MinCit Córdoba, a quien tengo el gusto de conocer personalmente desde hace muchos años y siempre se ha destacado por acompañar con mucho entusiasmo desde diferentes lugares de trabajo aquellos emprendimientos que estimulen la relación científica-tecnológica. Dada su amplia experiencia en estos temas, ha colaborado intensamente con la realización de estos espacios y en especial de este ciclo. En esta ocasión, que realizamos el segundo ciclo denominado Desde la mesada a la innovación tecnológica, el objetivo principal de este espacio es poner en marcha y proyectar una evolución hacia la vinculación y transferencia del conocimiento generado en las organizaciones de ciencia y tecnología por el sector socioproductivo. De esta forma se desea incentivar el diálogo, la interacción entre ambos sectores para permitir la detección de necesidades y oportunidades de vinculación y transferencia. Esto traerá como consecuencia la visibilización de acciones que desde las investigaciones en química orgánica pueden brindar soluciones y dar respuesta a los requerimientos y mejoras de la industria. Iniciaremos el segundo VOM con la conferencia del doctor Dreher, quien es investigador principal en la empresa Merck. A continuación habrá una mesa redonda con la participación de profesionales y empresarios insertos en diferentes industrias del área química. Ellos aportarán sus experiencias que darán lugar al intercambio de ideas y a la potencial implementación de nuevas herramientas que puedan mejorar al sistema actual de investigación, desarrollo e innovación. Antes de pasarle la palabra a Gabriel, quiero agradecer nuevamente la presencia de todos ustedes, agradecer también a nuestros auspiciantes cuyo aporte ha sido tan importante para nosotros y en forma especial a la comisión que se abocó a la preparación de este ciclo y que han trabajado con mucho esfuerzo para poder hacerlo realidad. Thank you very much, Professor Dreher, for your participation in this Congress. It is a great pleasure to have you as speaker today. Welcome to the second virtual opening meeting. Gabriel, todo tuyo.
Muchas gracias, Miriam. Realmente un placer. Bienvenidos a todos a este segundo ciclo virtual de conferencias. La verdad que siempre es un placer poder trabajar con este equipo y sobre todo desde el Ministerio de Ciencia y Tecnología de la provincia de Córdoba, agradecidos y en voz también de nuestro ministro Pablo de Chiara, quien hoy no pudo estar presente por cuestiones de agenda, ponemos en relevancia y damos la importancia que merece la disciplina de la química orgánica y el impacto que tiene la química orgánica y los profesionales formados en esta disciplina para la provincia de Córdoba. La química orgánica es una disciplina que atraviesa a diferentes ejes estratégicos que hemos definido conjuntamente con el sistema científico-tecnológico y con las empresas de Córdoba, por ejemplo, en lo que es agroalimentos, en biomateriales y en la misma biotecnología, en donde la química orgánica es base para el desarrollo de conocimientos, de tecnología y que sean luego transferidas al sistema socioproductivo. En ese sentido, también en este precongreso y trabajando fuertemente con el equipo de Miriam, vimos la importancia y la necesidad de poner en valor lo que es la vinculación tecnológica y poder acercar a los científicos, o sea, la mesada, con digamos, con la realidad o con las necesidades o cubrir necesidades y oportunidades que tiene el sistema socioproductivo, tanto de la provincia como de la región. Y ahí es donde entra la importancia también, como les decía, de la química orgánica para estas empresas de nuestro medio. Como ustedes saben, Córdoba es una gran generadora de biomasa, o sea, de material orgánico, y cómo transformamos y generamos valor a esa gran biomasa que tenemos en Córdoba. Y ahí, de nuevo, la química orgánica es base para esto, ¿no? Esta vinculación público-privada y la interrelación que hay con el sistema científico-tecnológico y el sistema socioproductivo es uno de los grandes objetivos que tenemos dentro de la Secretaría de Articulación Científica Tecnológica. Por ello que toda oportunidad o todo trabajo que se nos presenta para poder llevar adelante esta vinculación y generar espacios donde podamos sentar a investigadores, científicos y el ámbito gubernamental para la generación de políticas públicas que sean beneficiosas para lo que es el desarrollo económico de la provincia, ahí estamos presentes, apoyamos y trabajamos fuertemente. Así que bueno, simplemente agradecerles, desearles que disfruten de este espacio. Vamos a tener una mañana realmente con unos expositores y una mesa panel de primer nivel, en donde tienen que aprovechar esas experiencias, intercambiar ideas y llegar al debate para poder generar nuevas herramientas y poder darnos a nosotros también información y herramientas para poder generar las mejores políticas públicas para seguir creciendo en Córdoba y en toda la región. Así que muchísimas gracias y muy buena jornada para todos.
Bueno, muchísimas gracias, Gabriel, por tus palabras. Creo que sí, que va a ser una jornada muy productiva para todos. Esperemos que sea así. Y que va a ser un aporte importante para la 23ª edición de SINAQO. Así que bueno, le cedo la palabra a Natalia, que va a presentar al doctor Dreher, y comenzamos entonces con el ciclo. Muchas gracias nuevamente a todos.
Buenos días, good morning to everyone. So I will be presenting to Dr. Spencer Dreher, who was proposed as an invited speaker by Dr. Ana Belomo. We want all to thank you for that. So Dr. Dreher holds a PhD in organic chemistry with more than 20 years experience in process chemistry, drug development, and medicinal chemistry drug discovery, with expertise in high throughput experimentation and transition metal catalysis. His abilities to conceive and drive successful collaborative scientific efforts inside Merck and academic work externally with the University of Pennsylvania and Princeton University resulted in high quality publications. And so since 2015, Dr. Dreher has been the lead scientist in Merck Chemistry Capabilities and Screening Group in Discovery Chemistry at Merck, where he provides in-lab scientific leadership and innovation for discovery chemistry efforts to standardize HTE technologies beyond Merck. Also, he's a member of Journal of Organic Chemistry Editorial Advisory Board from the ACS, and today he will present his talk entitled HTE in Medicinal Chemistry. So we welcome Spencer to this meeting, and we hope that we had a lot of questions that all the audience can start putting on the chat. Yeah, and so at the end of the talk, I will be reading your questions. Yeah, so it's all the presentations now.
Thank you, Natalia. Buenos dias. That's the extent of my Spanish. Both of my children are learning to speak Spanish though, so that's a good step in the right direction. Let me get my screen set up. Okay. You can see my slides. Natalia?
Yes, it's okay.
Okay,
yeah,
great.
It's okay.
Thank you so much for the invitation today. I'm very excited to present some of the work that we're doing at Merck and with a lot of collaborators, as you'll see later, in academia to develop HTE as a tool for chemistry, for medicinal chemistry, for process chemistry, but also for academic chemistry. Hopefully we can show some things today that will inspire people to think about trying these tools for their research. So, as Natalia said, I work at Merck, and I've been involved in the entire sort of spectrum of synthesis that they have at Merck. So I started my career actually in process chemistry, which is at the end of the process in drug discovery. And then I've worked my way backwards all the way into medicinal chemistry during my career. And if you think about what process chemistry and medicinal chemistry do, in MedChem, they're trying to figure out what types of molecules to make to solve important biology problems. Okay, what if you have a disease, what kind of drug-like molecules do you need to make to impact the drug, the disease? And you use this design, make, test cycle to invent new molecules. In process chemistry, we try to figure out, once we have molecules that are known to be important in biology, we try to figure out how to make them on very large scale so that we can use them for clinical trials and also for manufacturing someday if something were to become a drug. So again, I worked in both of these capacities. I wanted to start by saying a very positive statement about the state of chemistry and catalysis. I think we're in an amazing age of innovation. Just endless amazing papers coming out in the literature from academic labs. I'm not going to go into these in detail, just to say that there's many new different types of catalysis happening in the literature. Photoredox catalysis is an amazing advance on using light to improve the state of catalysis. Lots of papers on using catalysis on very complicated biomolecules, and even using biomolecules, enzymes, to do chemistry in a very exciting way. So many different things that are coming out in the literature to impact how we make molecules. And what I'm going to be focusing on today is the way that we're thinking about how we can use new tools in order to use these technologies in the real world synthesis. So I'll just start by saying that we're very aware at Merck about the literature. We read all the papers, and we know sort of what the different transformations are that people use, and also sort of the best types of conditions that they use to solve problems. So in fact, in our labs, we have a document that we send to people that is called the Catalysis Selection Guide, and we try to put the best conditions so that everyone can see what the best ways to make molecules are from the literature. Having said this, the problem is that the molecules that we need to make and the molecules that are in the literature are very different. So these types of drug-like molecules, in order to be viable in biology, have lots of functionality that is actually very difficult to manage in a catalytic reaction. A lot of strongly binding nitrogens, for example, or NH bonds or OH bonds. So the physical properties of the molecules that we need to make are very different from the physical properties of the molecules that are in the literature. So you can see these types of compounds on the bottom here are drug intermediates, and you'd want to functionalize one of these molecules, say at this bromide with a catalytic reaction, to make libraries of new molecules using these compounds, and it's very difficult to get these compounds to work. And this is just another way of saying the same sort of thing. We did a study recently, and we showed that a lot of the sort of recent catalytic chemistry conditions that were published in the literature, if you map the structures onto drug-like space, you can see here that these are all the types of molecules that come into the publications in the literature. And this is the type of space that drug-like molecules fill up. And again, this is spread out in two-dimensional space based on the types of properties that molecules have in drug-like space. So we're really not covering the space that we need to cover in order to to make these types of molecules. So what are we going to do about it? We have really tried to develop new tools in HTE in order to solve these difficult problems. So, for example, the traditional process that you would use to solve a chemistry problem would be to read the literature and identify a paper that you think is the best, maybe looks the most like what you're trying to do, run that experiment. If that reaction doesn't work, you take another paper and you run that experiment. And what we try to do is to take all of the different reactions in the literature and run them all at the same time. That's a very simple way to think about HTE. So we're going to take, you know, all of the solvents and bases and catalysts that people have reported, and we're going to run them at the same time. We're also going to sometimes cross the conditions. For example, use a base from one paper and a solvent from another paper and a catalyst from another paper, and end up running a full factorial combination of those in order to create an array of reactions. So we actually like to think of the way that we started doing this work as 1980s biology. So it's kind of like having a small 96 well plate and handheld pipetters. And we would dose things into the wells. We were actually going to use this block on the bottom, which is all glass vials, but we would dose things into wells by hand and make combinations of things and run the chemistry this way. So this is the way we started. And I put this down here because this all started in process chemistry. We were trying to figure out how to enable catalysis to run reactions on very large scale. And we found very quickly that we could solve a lot of the types of problems that we would come across in trying to use difficult catalytic reactions, new reactions from the literature in drug discovery and development. So this graph shows you the percentage of times that we could find a solution to a problem when the initial literature conditions failed. So a chemist might go and try one set of conditions to start, and if it works, that's great. If it doesn't work, then they come to screen using our HTE tools, and we can solve the problem for different types of chemistries. So Suzuki reactions, Miura borylations, sp3, sp2, Suzuki couplings, etc., at a very high rate using these tools. We find something in our array that works. And you can see this last bar where we say other is all kinds of different chemistries that we would use. So asymmetric hydrogenation, almost anything you can imagine. We've tried to do it in this format, and we were able to do this. So again, this is at the beginning, all very focused in process chemistry, large scale. We want to take these results that we find in very small scale and run them eventually in very large scale. So that's where this started. At this point, I want to focus in on the use of HTE in medchem. So somewhere in about the middle of my career, I took an interest in trying to think about how we can impact medicinal chemistry with these same tools. And you'll see from this graph that if you look at our electronic notebooks, almost all of the reactions that people use, 80% of them are very simple reactions that they use over and over and over again, like amide bond formations or reductive aminations, right? Or those types of basic SNAR reactions, basic disconnections. Very few people were doing what you might call modern catalysis at all in medicinal chemistry. And if you look at the numbers of this 18% that we're doing them, most of them are sp2 Suzuki reactions, which are very robust reactions amongst catalytic reactions. So, very small amount of their effort was going into running reactions with all of the new reactions that were coming out of the literature, all of these amazing new possibilities. Very few of them were being used. And why is this? It's because medchem moves very fast. The substrates are very precious. A lot of times you're making, you want to make biological molecules with a very small amount of material that you have in your hand at any moment. And the timelines are very tight. So you're moving very fast and you don't have much material. So you're going to use your substrates to try to make as many compounds as you can which leads people to use easy chemistry. And those compounds will end up in biology for screening. So what we wanted to do was to see if we could come up with a way where we could take something that was, say, a 96 well plate approach and shrink it down into a smaller array, and also make it very easy for chemists to do in their labs. So what we did is we came up with this idea of a catalysis kit. And this kit would have the best conditions from the literature for any specific transformation. And we used our sort of internal knowledge about what reactions worked from a 96 array, focusing in on those we thought had the best chance of success. And one key thing about the kit is that we're actually going to dose catalysts and bases, for example, into the kit in advance. So a chemist can just take this kit that has everything in it and just add their substrates into the wells. And then they run these reactions, a small array. And we found that we could actually do a very good job of solving problems with a much smaller array. So 24 reactions instead of 96. We were pretty good at choosing 24 reactions that would be good to solve the problem. And I will say that this approach is now commercialized by Sigma Aldrich. So anyone who wants to try this, you can order these kits from Sigma Aldrich, and you can run these in your own labs. So it's a very easy way to get started with HTE. And just as an example here, we have a Suzuki reaction, and on the top, we have a very plain looking Suzuki reaction with this boronate here. And you can see that if you run this very easy reaction, almost every reaction works very, very well on our kit. So these are all very good conditions we have on here. You introduce a single nitrogen, everything else is the same into this system, and you basically get one hit out of this approach, right? So this shows you what we come across over and over and over again in catalysis, is that we run this sort of array of different catalysts. These are phosphine catalysts with different bases, and we find these very specific results to problems. Okay, so at this point, we started to think that this approach might add value in academia. And so we contacted the University of Pennsylvania, and we're very, very fortunate to get NSF funding to build this approach out. And the way that this worked is we got money from the government, and we were able to hire 4 very talented postdocs who came to Merck for a year to learn how to do what we did, and then they went back to Penn and set up an HTE lab at Penn. I was extremely fortunate to meet the very, very talented Dr. Anna Belomo in that process. She was very important in helping us build all these tools, and you're very, 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 very lucky to have her there. So we wanted to use these tools to see if we could impact academic research. One thing that we did in this process is we actually put a lot of effort into trying to figure out if we could go even smaller in scale in our reactions. So initially, almost all of our work was in these 1 ml vials, and we spent a lot of time trying to figure out if we could go even smaller in scale. Can we use, because again, if we're trying to impact medicinal chemistry, we need to run reactions on the smallest scale as we possibly can, so we're not wasting material. So we put a lot of effort into running reactions. What we did is we ran reactions at all 3 scales, at 8 ml vial scale, a 1 ml vial scale, and what we call a microvial scale. And we showed that the data for the reaction chemistry conditions that we ran in this format were very similar. So we could actually say that with good confidence that the chemistry that we would find in this scale would scale up well to a larger scale. This is a scale you might do a preparation on in medchem. You run a reaction at this scale, and then you purify it and you do biology. So this is the scale, this is the synthesis scale, and this is the screening scale. So we did this in collaboration with Penn. Another thing that we did is we wanted to just look at very fundamental disconnections that were important in drug discovery. And we actually made a list of reactions at the time. This is 2007. of reactions that we thought we really needed that didn't work well in our labs. And one of them was an sp2, sp3 Suzuki coupling, so an alkyl aerobond disconnection. It was something that was very poorly developed in the literature at that point. So using our HTE tools, and this is with Professor Gary Molander, we did a screen, and this is about 296 well plates with ligands and catalysts and bases and solvents. And we actually started with relatively difficult academic substrates. So a slightly hindered aerochloride as a starting material and an heterocycle as a starting material. And by starting at slightly more difficult substrates, it allowed us to figure out the conditions that we needed for catalysis relatively quickly. So we ran the screen. This is what a typical data set would look like. And you can see from this data that for the aerochloride in this coupling, sp2, sp3 coupling, there were quite a few hits that looked pretty good. But again, when we used the heterocycle, almost nothing worked, and we had one hit. And it turns out that this condition and this condition are the same. It allowed us to find very quickly this set of conditions, and we published this as one of the first papers in our collaboration. So this is something that just wasn't, it was a basic disconnection in catalysis that didn't have a good solution, and we found a solution very quickly. And I think the most exciting thing about, and I'll just say that we did actually write about 25 papers in this collaboration doing sp2, sp3 type disconnections with the professors at Penn. So one of the most exciting things about this collaboration, though, was leaving a really high quality HTE lab at Penn. And so that, you can go on the web and you can see what they have in terms of how they built the lab out. This was NSF funded. The students at the time, and Anna was one of them, wrote a nice review on using HTE in a sort of scientist-led approach. So it was an idea where we trained these guys originally, they trained additional people, and pretty soon almost everyone at Penn knew how to do HTE. In addition, as part of this, we spent a lot of time and effort trying to make sure all of the technologies that we were developing became commercially available. So there's this company called Analytical Sales and Services where you can order all of the equipment that we use. And we've worked with them to build out all of the new things that we're doing, because we've come a long way from here, and that's kind of where the rest of my talk is going to be, is sort of where have we gone with HTE since this work. So just to end the first part, though, we've now worked with many different professors at universities across America and even a little bit around the world, Spain, to try to develop these tools for academic purposes. Okay, so if you think about chemistry, our goal is to run things in metric tons in manufacturing. Right. If we get a successful drug synthesis, we want to be able to run reactions on massive scale. But if you think about our experimentation process, a lot of times we run test reactions at this scale in order to scale up to metric tons. We've just talked through how we can scale down from sort of a 30 milligram reaction to about a 1 milligram reaction for HTE. But we began to wonder, can we go smaller than this? Even this scale here, which we're using, is 30 quadrillion molecules, right? This is 30 quadrillion pennies compared to the Empire State Building. Do you really need to waste 30 quadrillion molecules to test whether reaction works or not? Okay, so as global stewards, can we do better than this? As chemists, can we go smaller scale? So we'd like to think, can we do chemistry in a very, very much smaller format? So the answer is yes, and it took us time to figure this out. What we realized is that biology does their science in very, very small scale already. So what can we learn from how they do biology? So borrowing from some of the things that they do in biology, we need to run reactions in high boiling solvents. right? You can't run a very, very small scale reaction in THF, for example. You're going to have to use automation if you're going to do this, because you're not going to be able to dose reactions into a plate this size with your hands anymore. We need to use cheap plastic plates because, you know, you can't really use a glass plate at this scale anymore. How are you going to analyze all these reactions? And, you know, once you run all of the reactions, how are you going to deal with all the data that you generate? So just another way of thinking about this, you know, chemistry typically is run in sort of lower boiling reactions as heterogeneous mixtures at high temperature. Most catalysis is run this way. Okay, can we actually change the methods to work in homogeneous solvents in a high boiling solvent at low temperature? If we can do that, then we could think about doing chemistry in this format. So we had a very talented postdoc in the Merck postdoc program named Alexander Pietragos-Santania, and he had the wonderful idea of exploring organic superbases to do catalysis. And what does this mean? One of the key problems with catalysis in terms of trying to think about how to miniaturize it is that most reactions are run with metal bases. So either T-butoxide or HMDS bases or potassium carbonate, cesium carbonate, etc. And these are very hard to think about how to put into a very small vial because they're solids and they're insoluble in reaction mixtures. Organic superbases would be an ideal idea to do catalysis because they have the right strength in terms of their pKa, but they're also hindered so that they don't cause a lot of trouble that these bases can cause in like HMDS bases, T-butoxide bases, where they can actually be very hard on reactions in terms of stability, but also they're very soluble in high boiling solvents. So Alexander had the idea of using these bases, and we were able to figure out how to do many different types of cross couplings in DMSO in sort of a plastic plate format. So very quickly, when we learned about this kind of approach, discovered this approach, we did a comparison of running reactions in a 1 ml vial, which was our smallest, in our micro vials, excuse me, this scale, and comparing that to what the results we got in a plastic 1536 well plate running reactions at 1 microliter scale. And you can see that the data that was generated is very similar in terms of the hits that we found. Okay, so this is using organic superbases, catalysts, and in this case, a CN coupling reaction. The reactions were all dosed using this mosquito robot, which is an amazing piece of equipment that where you can pick up nanoliter size drops of materials and dose them very accurately into wells. So we can dose reactions at 1 microliter scale by combining different reagents using this mosquito robot. And you can see here, this is what a 1536 well plate of data looks like. And in this screen, you can see we have a whole bunch of different types of nucleophiles that we're coupling. So CN couplings, sulfonamide NH couplings, and heterocycle couplings, but we can also do CO couplings and phenol couplings and CS couplings, Suzuki's and active ester couplings, Heck reactions, Sonogashira reactions. So we found we could do all of these different types of reactions in this format just using organic superbases and catalysts, sort of Buchwald type catalysts. And we, almost all of these couplings we can do in this format. So for us, the most exciting part though is when we started looking at real drug-like molecules in this format. So if you take these types of molecules that we talked about before, and you run them with the sort of typical highly polar molecules that you use in drug discovery, we were able to use this approach to find hits for many of these types of compounds. So we could find a base and a catalyst combination using DMSO solvent in this format, and then we could scale those reaction conditions up to medchem scale to make material. Not only can we screen bases and catalysts, in this case, you can also screen amounts of reagents, so what we call just continuous variables. So you can look at different base and catalyst amounts in this format, and this allows us to solve some of the problems in this small screen down here that we weren't able to solve in just base and solvent catalysts. Okay, so I'm coming close to my end. Just a couple more slides about where we're going. Ideally, we would like to not have to screen at all. And so it works, but it's very time consuming. It's also expensive. You need a lot of equipment to do this kind of work. So ideally, what we'd like to do is we'd like to generate data that we can then use to predict what conditions will work for very specific types of couplings. And people have been talking about predicting reaction outcomes for a long time. The problem is that there's no good data out there. If you look at the literature, as we talked about before, the types of problems that people are trying to solve don't look like the problems that we are trying to solve. So you have to generate data that is relevant to the problems that we're solving. So we feel like with this sort of nanoscale chemistry, with robotics, that we can actually generate complete data sets where we have the success and failure. That's another problem. The literature only shows success. It doesn't show you the failures. We can run reactions under highly controlled conditions. We can use relevant substrates, and we can run reactions on the types of problems that we're having in the real world. And this idea is something that's gotten a lot of attention for us recently with the government, with Google and other sort of entities who want to learn how to generate data like this for machine learning to predict reaction outcomes. And we just, about a couple of years ago, published our first paper in this area. I'm not going to describe this experiment. It's a little bit complicated, but I'll just say that you can run chemistry in our nanochemistry format. You can run, in this case, we ran about 5,000 experiments, which is 3 plates of data here. And you can use that data to predict reaction outcome. And so we're able to get a reasonable R-squared predictivity in this sort of approach. So that's sort of a split sample size where you take some of the data and see if you can model the rest of the data from that approach. That's how they typically do machine learning. Interestingly, we were able to calculate the properties of the molecules that we were looking at in our screen and use those predictive properties to actually predict reaction outcome, right? So you didn't maybe have to run the reactions if you can predict the properties of those molecules. And finally, we spent a lot of effort trying to understand the results that we got from this and boil it down into mechanistic analysis that we could use to sort of forward our understanding of the problem. So this is something that is definitely a future direction for catalysis. We know that applying catalysis to real world drug-like problems is very difficult. We know screening works, but we would really like to be able to predict instead of having to run all of the experiments in screening. I'm going to leave you with one more very interesting idea that we're working on in our labs, and that's this idea that can we actually run chemistry and biology at the same time? In other words, can we run a screen in a 1536 well plate and find the reactions at work and get biology on that same sample without ever having to scale up? So we've actually had good luck at trying this with certain types of chemistry. So you can run a plate of reactions, you can find the best conditions, and you can take the sample right out of that well and put it into a biological assay. And I'm not going to go through this, but just to say that it's been very effective in many different types of biological assays. We have to do obviously a lot of control experiments and really making sure we understand the impact of the chemistry on the biology, because we're not purifying these reactions. We're taking them as they are in the wells and running them in biology. I will say that this works very well for a lot of different types of simple reactions. Again, amide bond couplings, SNAR, et cetera. And really the challenge for us is to try to figure out how to do this with catalysis. Can we use palladium catalyzed reactions, copper catalyzed reactions, and do biology on those samples directly? And the answer is yes, we can, but not always. And we need to learn more over time on how to do this. But this represents a very exciting new direction for chemistry and biology, because you can run reactions on very small scale quickly, and you can do the biology at the same time. So my last slide is just to say that there is a very modern version of chemistry that we're working on developing. So using computationally driven library design. So we're using all the tools that are being developed in high-end computing to design libraries of molecules. We're using machine learning driven synthesis in our nanochemistry platform to find the right conditions to make the molecules we need to make. And then we're increasingly trying to use integrated sort of direct to biology chemistry from our small scale to be able to do medicinal chemistry. So this is kind of our story of HTE at Merck, starting from process chemistry and going smaller and smaller and smaller, all the way into medicinal chemistry. And I want to thank you for your attention. I'd be happy to take any questions.
Thanks, Spencer, for the great lecture. So I have a couple of questions that people were sending by the chat.
Sure.
So I will start Fabricio asked, as you showed at the beginning of your lecture, virtually any organic transformation can be catalyzed by an artificial metal enzyme, as those developed by Thomas Ward, Gerald Ruffles, and other teams. My question is, if you consider that those state-of-the-art biocatalysis reactions can be thought as key reactions in miniaturized settings, or some more research must be done in this line?
I understood the first 3/4 of your question. Could you repeat the last part, please?
Okay, so if you consider that those state-of-the-art biocatalysis reactions can be thought as key reactions in miniaturized settings, or some more research must be done in this line?
I think that that's one of the most fascinating advances in chemistry, to be able to do catalysis in an enzyme. It's very difficult to do at this point. You have to sort of engineer the enzyme to have the right functionality in it to be able to do sort of chemocatalysis. There's only a few examples of it in the literature so far. It's so difficult, very few people are doing it. But the amazing part of it is that once you learn how to do it, especially for things like enantioselective reactions, then you can just evolve your enzyme. All you need is a glimmer of a hit, right? Where you get, enzymes are almost always enantioselective when they work. But if you get even a smallest amount of product out of a reaction, then you can evolve the enzyme to better and better activity and reactivity. So I think it's one of the most exciting areas in chemistry, but very, very difficult. So we're doing it, but we're not doing it enough at Merck.
Okay, so it's work to do on that side.
Lots to do.
Ana Velomo, she asked, what do you think that research in academia is not covering the medicinal chemistry space needed? How we can change that?
I think a lot of the way to impact academic research is about having access to funding, because you need equipment to do this kind of work. I think the thing I haven't talked about is analytical. And a lot of the cost is analytical, and I can talk about that if people would like to know more about that. to do the chemistry is very easy. It's and relatively inexpensive. You can buy the aluminum plates and the glass vials and stir bars and things like that relatively cheaply. The analytical is expensive. So we need to do the work we we have about 10 UPLC-MSs in a room, you know, to analyze all these reactions, and that's $2 or $3 million, right, to do that. For an academic lab like Penn, they have a couple of UPLC-MSs. They got money from the government, so I think the schools need to, you know, professors at schools or even regional schools in the same region need to come together, write proposals, and get some of the equipment that's needed, especially analytical. You can do things like high throughput TLC, not too hard to spot plates and do TLC. So you could run a 24-well reaction to see if you get new spots, and then take that reaction and maybe run an NMR on it or something. Those things can be done, but certainly it makes it a lot harder to do. So to answer Anna's question, I think really it's about spreading access to the tools. And I think, you know, I understand that not everyone will have a large amount of money accessible, but maybe if you think like think together, right, that multiple professors or multiple schools can write grants to get money, that you might be able to do that. And I think one thing that worked really well at Penn is that they would run screens for other schools. So if they have the technology, they would collaborate with professors at other schools and run the chemistry for them and just provide them with the results. And that was something that was very successful. So that's one way to go. If you had one institute that started doing this in Argentina, others could take advantage of it. Okay.
So another question is can you describe how you analyze rapidly or fast 15 and 36 reactions? Right. So it's like a lot of reactions to analyze. So.
I have my backup slide here, and there we go. So as I mentioned, and I don't have it on this slide, but I can just verbalize it. When you run a 1536 well plate, it takes about 4 days of UPLC-MS time. So UPLC-MS is a 2-minute HPLC assay. So it takes 4 days to run that. So clearly that's very slow and expensive too, to do it that way. We've tried to invent new ways of doing the analysis that can be helpful. So in our original nanochemistry paper, we use what's called miser. And what we're really doing in a miser experiment is just shooting the crude samples into a mass spec, and we're looking for a mass signal. And we found by doing this that we could actually see which reactions have some product in it. It doesn't necessarily give you a very good quantification, but it tells you which samples to look at. So then you can really cut down the number of samples you have to run on UPLC-MS. We did find that you could also put different reactions that have different product masses in the same well. So a miser would take about 9 hours, from 4 days to 9 hours. If we pool the samples that have different masses, we could actually run the whole thing in just over 2 hours. So that's good. You could find your hits that way. We're really working on what we call ultra high throughput chemistry analysis. And ultra high throughput analysis, we're saying, is less than 1 second per sample. And we did write a paper recently using MALDI, which is an approach where you spot all your samples on a metal plate and you shoot lasers at it. And you pretty much, you can analyze as fast as the laser can move. So it's very fast. It moves across, you know, like this. And it also just gives you a mass signal. It shoots the sample kind of comes up into the mass spec, and it tells you whether there's product there or not. And this is something that if you really want to do this 1536 approach, is very valuable to being able to do this. Again, a very expensive approach that not everyone will have access to for sure. But I believe Penn actually has a MALDI instrument, so it is something that academic labs can start to consider using. So the real answer, there's no practical way to do it. You have to have a lot of expensive equipment.
I understand. Yeah, really expensive problem. So
we would love to have a light-based approach or something like that, right? So obviously in biology they use light. That's how they do it. They have an assay that will show you based on UV, based on the light that you get from the biological assay. It's hard to do that in chemistry, though.
Thanks. So another question from Marcelo Puyati is that maybe it's a possibility that heterogeneous catalysts are helpful in the activity test without purifying the sample, without the need. If you don't need to purify that, you can use maybe heterogeneous catalysts.
Right. Yep. I presented this story in a very simple way. We have used even our nanochemistry with more and more heterogeneous types of catalysis. We've learned how to dose solids and things into these wells. Heterogeneous catalysts, but also heterogeneous reagents can be very valuable for getting pure samples. Absolutely. And we haven't done a lot of, say, bead chemistry on beads and things like that in this format yet. But that would be a great way to run a chemistry reaction and just take the liquid and do your biology. What we do right now is something called SPE, solid phase extraction. So we can take a plate and we can pass it onto a resin, and that'll catch our substrates, our products, and then we can elute a lot of the garbage away, a lot of the chemical reagents away, and then elute off our product. That's sort of the same idea. That's something that works pretty well to able to get at least a more pure sample. You don't get, you're not going to remove similar molecules that look similar to your product, side products and things like that, but you can remove catalysts and bases and things like that. So SPE is another way to do that.
Okay.
We're finding we don't need to use it that much though, that the direct biology actually works pretty well a lot of the time. It's very surprising. We didn't think it would be possible to do that.
Thanks. And I will have like a last questions before continuing with the schedule that we have. So Bruno Gonzalez Nunez, he asked, okay, he thanks for your talk, and he asked if you have experience running HTE experiments with complex structure like natural products and substrates.
So yeah, we're not quite as complicated as natural products. We certainly are doing lots of large molecule HTE, so proteins and what do you call it? Sort of large ring molecules that look somewhat like natural products. Molecular weight 1,100, 1,200 molecules. All kinds of things with sort of slightly larger than small molecule array. Things like targeted protein degradation. These are new sort of areas in biology and chemical biology. I know that there are some professors who are starting to do natural product synthesis in this array. We haven't done that ourselves. We used to have a natural products group at Merck, but we don't anymore. So. Okay.
So we don't have any more time for more questions, but there are still some of them. So sorry to the people that I didn't mention your questions, but thank you again for your wonderful talk. And we really thank you and all the audience for being here. So we'll continue now with the rest of the the round table. And thank you again. Yeah.
Thank you so much for the invitation. And I think you have my email. So if anyone wants to have any follow-up questions, feel free. And I can have my kids translate for me if you want to write it in Spanish.
Okay. So we put on the chat your email. Yeah.
Okay. Take care, guys. Thank you so much. Bye.
Bye.